Develop a plug-and-play platform to build functionalized synthetic cell building blocks. My name is Ariory, and I am from Mississippi State University, and I am currently an RU student here at the University of Florida. Some things about me is I love to eat, travel, and for my downtime, I like to look at different languages and see if I can learn a few. Summer, I'm currently working with Dr. Carl Denard in his lab at the UF Chemical Engineering Building. Dr. Denard is my faculty advisor, while Luis is my PhD student mentor here, and I'm very thankful for both of them. The rest of Dr. Denard's lab, and I am very thankful to each and every one of them. I've learned so much from all of them, and I am so thankful that they let me come and join and put up with me with all my crazy questions and made me feel like I belong. Even though I'm just here for the summer, I will never forget y'all. Thank you so much. One form of life. Through countless of different living organisms in our world, it could all be traced down to a single cell. With the cell not having many differences from these different organisms, this will give us a lot of leeway to further understand its full functions. It is really fascinating that something so small could be so complex in replicating its function. Some general cell functions include genetic information storage and transmission, protein synthesis, energy production, metabolism, reproduction, cellular communication, and signaling. Synthetic cells are introduced for us to be able to control and minimize the complexity of a cell in a very simple term. As you can see on the image, this is an example of a cell membrane that has a function within it and a signaling that is actually provided on the edge of it. There are usually two approaches in making synthetic cells, which are top-down method, which provides a bigger picture. In a sense, this is where you take part of a complex structure and you break it down to its most simplest term. While the bottom-up process is starting from like the very simplest term and you make it more complex as you add more things to it. Bottom-up process is what we will be using over this summer. In a sense, if there was like a mistake within the experiment, we could use a prior knowledge to fix the issue at hand. So we will be able to create what we want and understand it in a very broad way. So the way we start this bottom-up strategy is using a cell membrane. So previous experiments, they have used lipids, polymers, and global proteins to make the structure. We are using global proteins to make this vesicle due to its compatibility, and they are stable, permeable, and they have self-assembly capabilities. That's important part of a vesicle that will be targeted throughout the experiment. Um, Studies before have actually used global proteins vesicle, and it has been reported and it was actually characterized. So this method should be able to work in our sense. So once the vesicles are fully made, we are going to be able to incorporate a new function into the vesicle, and one example of it could be sensing. So this image that recently just popped up is just another example of the cell membrane having actuation within the whole cell membrane, and that is like a representation of its function, while the lining of it has sensing, so it could actually sense what's happening around it. And this is just another image that shows two proteins that could make vesicles, which is an M cherry and a ELP. This summer's goal was to optimize the protein purification process and develop a protocol for the tri-block building block assembly. The mid slash long term aim is to imbue these global protein vesicles with properties that mimic cellular functions such as sensing, actuation, and self-maintenance. The previous slides, the protein that were shown will fuse into this building block, for example, the M cherry and the ELPs. So the typical structure of a vesicle is an amphiphile, which consists of a bottom part being hydrophilic, which is the ELP, and the top part being hydrophobic, which is the zipper domain and globule. This building block structure has been previously developed by other individuals. It serves a singular function, making it a fundamental component. It provides a solid foundation for further experimentation and new developments. However, an exciting possibility lies in enhancing this block by introducing another protein, thereby enabling it to perform two distinct functions simultaneously. This addition will significantly expand its capabilities and potential, and this will be made possible by actually implanting a protein at the end of the tail of this protein. So this is a possible new structure that we are hoping to develop. So this schematic illustrates the desired structure of the tri-block vesicle that we are looking for. The main objective is to incorporate an additional function by attaching our protein of interest at the end of the vesicle, which is the purple image. This could be a fluorescent protein or any protein that you may want to do. The success of this tri-block system relies on a robust bioconjugation between the spy catcher and spy tag. This is all started with the plasmid design and its construction, protein expression, protein purification, so that you can isolate the protein that you want. While that is made, you can finally do the vesicle. You can actually make your vesicle and find out its biophysical characterization. So this is important so that you can actually figure out the diameter, stability, and the fluidity characteristics of the vesicle and the design principle. So this summer I was heavily focused on the protein expression, protein purification, and physical formation. Within all these steps are what I was mostly involved in. So this is the process of the protein purification. So you start off with expressing the protein. Once the protein has been expressed, at the bottom of the bottle should be a collection of your cells. And why it is green? This is the reason because we are using a GFP. So it'll emit a green color. And everything else in the picture is just supernatant. And after we centrifuge it, this is typically the result. This part of the process is we're using the sonicator to break apart the cells within their solution, and we are using a nickel agarose beads to incubate it within the solution, and it should bind to the protein of interest that we want. And after we centrifuge it, that is what the picture is showing, where you can see the pellet, which is something that we don't need, and we're just keeping the supernatant, and that is why it's colored green, because we are using GFP once again. So this is the process of chromatography, where we use a gravity well to actually elute the protein that we want, and we put it into this tubes, and we collect samples to check their concentration. Once we collected all the concentration of our elution, we transfer it into a snake skin, and we put it into a 1 liter beaker and let it dialyze. Um, this is important because the elution product needs to be dialyzed so that the excess salt could actually be removed that triggers elution after dialysis. And then we use the SDS page to evaluate the protein purity of it. This is the optimization of our SpyCatcher ELP GFT purification. Recognizing the shortcomings of the previous protocol, we enhance its efficiency and precision, yielding higher purity and productivity in our future experiment. With the change of the histidine tag, this allowed the protein of interest to bind more to the bead so that it could actually connect with the proteins with a longer histidine tag. This also increased the imidazole concentration wash buffer so that we could actually wash more undesired protein from binding to the beads. We did a lot more elution steps. We started with 1 ml each time and incubated for 5 minutes and spun it again for 5 minutes. This allowed the illusion to actually bind to the protein. So and help time for the beads to actually bind to it as well, so more time can actually work for it. We added another millimeter of agarose beads because we found out that after we did all the steps, some of the proteins that we want was actually being thrown away. So we added more beads to actually compensate for that. So as you can see on the first graph, the bed volume, once we started with the 1 milliliter, but the culture media was just an 8, while adding more beads to it, we actually saw like a twofold increase, which is like really good. And the next image, the SDS page, shows on the second column, it's just that's the wash step. So that's why you can see like not a lot of not a lot of protein in there. While the fifth column, I mean the fourth column is actually showing a lot more protein in it. So this means we could actually maybe increase the milliliter of the bed volume again, and the spike catch or LPGFP is on the very last row. So you can see how pure our protein is after we did all this new optimization to it. The optimization is ZR spike site purification. This step was important because when they dialyzed the protein, it was aggregating since the water was used, which takes the salt out of the snake skin and the protein needs salt to form. So changing the buffer to the sodium chloride enters, the aggregation actually decreased, and sometimes it showed no aggregation at all. So on this SDS page, as you can see at the bottom of the paper, the second column is a spycatcher or EPGFP, while the third and fourth column are ligations. And the fifth column is a ZR spy tag. So the third and fourth column just shows a ligation product, and it's just to show that it could actually be modified from the third to the fourth, and this just shows the result that the ligation actually worked. With the third column, this is a 1-to-1 ratio, while the fourth column is a 1-to-2 ratio, because it seems that we found out that there was like not a lot of ZR spy tag was actually used on the third column, so an excess was actually used for the fourth column for it to show these new bands. This shows the vesicle self-assembly. So on the first image, this is just a control of the M cherry ZEZR ELP with a molar ratio of 1 to 20 with a 50 micromolar ZR ELP. So letter B, this shows that vesicles was actually formed. So as you can see, this very bright light, which is the green part, it's a vesicle, while C does not have any because the wavelength was there was only red dot because the red wavelength was used, which could mean the green dot from B figure the vesicle is hollow, which means that it doesn't contain the M cherry ZE. On D, you can see there may be a lot of coacervates, and the big green right in the middle at the bottom is also another vesicle that has M cherry and GFP in the membrane. But if you look closely and if it's like zoomed in more further, the other green spectacles on picture D may also be vesicles. While E, this is just a really cool picture that was taken, and it was like hypothesized that this may be just a lot of debris, so that's why it looks like this. F is a profile of a vesicle that was drawn to show that the vesicle was actually um, gotten there. So typically a vesicle, when the pattern has been drawn, there's this like big trench which shows like a vesicle, but a coacervate actually looks pretty similar, but the trench won't be as deep if it was a coacervate. It is also and there's it also doesn't have a zigzag formation at the bottom, which means it's not a mixture of vesicle and a quasarvate. So it can't be really determined if it's not, because further analyzation of this needs to be known much more. Thank you so much, Luis and Dr. Denard and the rest of my lab family. I will miss y'all. It's been amazing meeting y'all and being part of the family. This is a great memory that I will never forget. Thank you for putting up with me. To all the people that I met here,
crazy, funny, smart. I will truly miss y'all. I don't know what to say. Thank you. Let's say goodbye one more time. The Gator Way.